A very good morning to all of you. Today we are going to do microbiology MCQs. Microbiology is a very important subject. About 25 questions of the NEET PG come from microbiology. So if you do these questions correct, you will get full marks, about 100 marks just from microbiology itself. And some questions in medicine are also asked from the infectious disease part. Some parts of the parasitology are asked in medicine also. So let us begin with the first question. Draughtsman colonies are seen with A. Anthrax, B. Pneumococci, C. Pertussis, D. Yersinia. What do you think is the correct answer? Here you can see from the image clearly. They are caramcoin appearance colonies in which the organism gives this pattern on the culture media. Here you, why this pattern? What is the reason of this pattern? Well, this is due to alpha hemolysis. Alpha hemolysis. On further incubation, when you incubate it further, they become flat with raised edges and central ambonation. Flat with raised edges and central ambonation like a carom coin. This is known as a Draughtsman or carom coin appearance. It is seen in pneumococci. So the correct answer here is pneumococci. Let us move on to the next question. Granulomatosis infantiseptica is caused by Granulomatosis infantiseptica is caused by A. Pseudomonas, B. Chlamydia trachomatis, C. Group D streptococci, D. Listeria. Well, this granulomatosis infantiseptica is an intrauterine infection of the newborn. It is caused by Listeria. The predominant feature of this condition is extensive focal necrosis, affecting especially the liver and less often the lungs. Lung and liver necrosis occurs here. The mortality rate is very high mortality. Occasionally, meningitis may be associated with that. The organism can be isolated from the affected areas in the child and frequently from the genital tract of mother, who may or may not manifest the disease. So the correct option here is Listeria. Stalactite growth in ghee broth. You can see from the image clearly it is a stalactite growth. It is a ghee growth. It is due to the following organism. A. Yersinia pestis. B. Treponema pallidum. C. Haemophilus influenzae. D. Corynebacterium diphtheria. Stalactite growth in ghee broth. So, what are the cultural characteristics? It is due to Yersinia pestis. The cultural characteristic of Yersinia pestis is when you grow it in a flask of broth with oil or ghee floated on the top. A characteristic growth which hangs down into the broth from the surface, resembling stalactite. On nutrient agar colonies are small, delicate, transparent disc which become opaque on continued incubation. Then coming to blood agar, on blood agar the colonies are dark brown due to absorption of hemin pigment. On macaronchi agar, colorless colonies are formed. In broth, a flocculent growth occurs at the bottom and along the sides of the tube. So once more, the cultural characteristic of Yersinia pestis. When you grow in a flask of broth with oil, there will you see stalactite growth. On nutrient agar, colonies are small, delicate, transparent disc. On blood agar, colonies are brown due to deposition of absorption of hemin pigment. On McConkey agar, colorless colonies. In broth, a flocculent growth occurs at the bottom and along the sides of the tube. So the correct option here is Yersinia pestis. Milk ring test. Again, this is a most often repeated. Is detect which organism present in milk. A. Bordetella, B. Brucellosis, C. Bartonella, D. Salmonella. Milk ring test. Milk ring test is seen in brucellosis. For detection of brucella in infected animals, pooled milk sample may be tested for bacilli by culture and for antibodies by several techniques. In the milk ring test, a sample of whole milk is mixed well with a drop of stained brucella antigen. and incubated in water bath at 70 degree for 40 to 50 minutes. If antibodies are present in milk, the bacilli are agglutinated and rise with cream to form a blue ring at the top, leaving the milk unstained. If antibodies are absent, no color ring is formed and the milk remains uniformly blue. So it is brucellosis. Super antigen causes A. Polyclonal activation of T cells, B. Stimulation of B cells, and C. Enhancement of phagocytosis. The correct option here is polyclonal activation of T cells. You can click the link in the description box to join the 2-day ultimate Neet PG masterclass. It is free and it will help you to improve your performance in the Neet PG exam. It is a clear blueprint. So please click the link in the description box. Respiratory syncytial virus causes all. A. Coryza in kids, B. ARDS, C. Bronchitis, D. Common cold, except. Since this ARDS is not caused by respiratory syncytial virus. Microorganism which cause pyogenic osteomyelitis is A. Streptococcus, B. Staph aureus, C. Corynebacterium, D. Neisseria gonorrhoeae. Pyogenic osteomyelitis is caused by Staph aureus. In nutrient agar, concentration of agar is A 1%, B 1.5%, C 3%, D 4%. It is 1.5%. Which cells cause rosette formation with sheep RBCs? They are the T cells which cause the rosette formation with the sheep RBCs. Which is found in Dyggve syndrome? Well, this is a very important question. Dyggve syndrome. A. Tetany, B. Eczema, C. Abscess, total absence of T cells, C. Abscess, B cells, T cells. Now, Dyggve syndrome is an example of T cell deficiency that results from failure of development of third and fourth pharyngeal pouches. The clinical feature is enhanced susceptibility to viral and fungal and bacterial infection, especially mucocutaneous candidiasis. Facial abnormalities, hypertelorism, abnormal ears, short philtrum and micronathia. Hypocalcemic tetany due to failure of parathyroid development. Congenital heart disease, example Fallot pathology. Absence of normal thymus. Serum immunoglobulin concentration are frequently normal, but antibody response, particularly Ig and IgG, are usually impaired. T cell levels are reduced, not total absence. B cell levels are normal. Tetany. Complement formed in liver are A, C2, C4, B, C3, C6, C9, B, C, C5, C8, D, C1, D, C3, C6, C9. Hereditary angioneuritic edema is due to A. Deficiency of C1 inhibitor, deficiency of NADPH oxidase, C. Deficiency of MPO, D. Deficiency of bromoridine. There is deficiency of C1 inhibitor. Opsonization takes place through A, B, C3B. Staph aureus causes A. Erythema chancroid, B. C. Acne vulgaris, D. Bullous impetigo. It causes bullous impetigo. Which group of streptococci grow at 60 degrees centigrade? They are heat stable enterococci, that is group D. Fish tank granuloma is caused by M. marinum. M. marinum causes fish tank granuloma. Correct answer is C. Boutonniere fever is caused by Rickettsia connori. Which of the following is called praegenocard bacilli? It is Corynebacterium pseudotuberculosis, which is called as praegenocard bacillus. Darting motility occurs in which occurs in Vibrio cholerae, also found in it is also found in Campylobacter jejuni. XDR-TB is defined as resistance to fluoroquinolones plus ionized plus rifampicin plus uric acid. Hebranosis is caused by fish bacillus. Duodenal aspirate is used in diagnosis of Giardia lamblia. Izumi fever is caused by Yersinia pseudotuberculosis. Unsegmented eggs are seen in which parasite? It is seen in Fisheries tissue. Brazilian purpuric fever is caused by Haemophilus aegypticus.